And that's a great segue to introduce Dr. Patrick Joseph, who has been our resident go-to person on COVID over these last 6, 7, almost 8 months now. We so appreciate everything you bring to us. I'm looking forward to this morning's message. So, Dr. Joseph, it's all yours.
Thank you. Thank you. Let's see if we can pull up the slides. Yep. Very good. Thank you. Well, we're going to, these are the topics that I thought we might discuss today. A little bit about a change in a definition. There has been a lot in the news about reinfection and vaccine. And typically what I try to do is sit down and think about the questions that I've had since our last meeting and address those. And I use that as a meter of things of public interest. So the first thing is that 2 days ago, there was a change in the definition of a close contact. Now, let me first say that this is important for contact tracing. This is what is used if an individual is concerned that he or she may have been exposed to someone who has become positive. So this is important at the workplace, at schools, perhaps with acquaintances. When an individual calls you or a contact tracer calls you and says, you may have been exposed to someone who is now diagnosed. Previously, the definition was with less than 6 feet, with or without a mask, for 15 consecutive minutes. So a long conversation with someone at close range. With some new information from schools, this has been changed to 15 cumulative minutes. So at the workplace, this becomes important. If you're talking to someone for 5 minutes, 3 or 4 times a day at a range of less than 6 feet, and that individual turns out to be positive, that meets the new definition of a close contact. And as you can see from the top of the slide, this is only 2 days old. So again, this really applies mostly to workplaces or acquaintances if you are notified of someone that you may have seen, may have talked to, or know who has been diagnosed. Now, I'm using this slide to introduce the concept of reinfection. This is an artist's sketch of the coronavirus with the RNA inside the shell. And what's in the hand tweezers is a very, very tiny portion of the nucleic acid, a very tiny portion of the RNA that is used for diagnostic testing. So when we talk about doing a PCR testing, we're really only looking at this tiny little portion of a very, very long strand of RNA. And this tiny little portion has been selected because it's common to all strains of the coronavirus. However, all strains are not the same. And the nucleic acid sequencing here is really different among many strains. So far, the changes in the sequence only define the strains, but they have not yet defined that one strain is more infectious than another. Now, the value of looking at these long strands is to tell us where the strain came from. So, for example, the coronavirus cases in the West Coast came from Asia. The coronavirus cases that caused the hotspot in New York City and on the Eastern Seaboard came from Europe. So it's pretty easy to map the origin of this by looking at the nucleic acid. So what has happened is that the news has reported that selected individuals have been infected twice. And that's new. And the way that has to be proven is that the virus from the first strain is sequenced. That's looking at the nucleic acid sequence. And then an individual gets well or recovers and then gets a second infection. And for it to be a reinfection rather than a persistent infection, it has to be a different strain. And indeed, that has happened. And let me give you an idea of how commonly it happens, or probably better said, how uncommonly. In the United States, or in the world today, we have approximately 41.5 million cases, and there are 5 individuals who have a proven reinfection. And I will suggest to you that this is clearly an underestimation. There likely are more than 5, but it is going to remain very rare. In the United States, where we have about 8.5 million infections, there has been one person proven to have a reinfection, that an individual who was infected once, but then got infected again with a second strain. That was an individual from Nevada. And then you can see there are 4 other countries where there has been one case. Now, if you imagine to prove a reinfection really means having access to the virus from the first infection and comparing it to the second one. And that is quite difficult. So I'm going to suggest to you that this still remains a rare event with only 5 proven cases, probably even more rare than individuals who get chickenpox twice, or individuals who may get measles twice. That is rare. This is probably even more rare, but we want to understand that it can occur. Because of its rarity, there's no change in the current guidelines, thinking, or discussions, meaning that once a person is infected, he or she is likely immune for at least 90 days. This just shows the rarity of an exception, but no changes that immunity probably exists after infection for at least 90 days. And as time goes on, all of us, of course, are hoping we can extend that comment, extend that axiom, hopefully for 120 days and continuing on and on and on. So that's where we are with reinfections. You should know about it, but nothing to worry about. Now, an update on the vaccine. This is where we are as of this month. There are 11 phase 3 trials going on in the world. These are thousands and thousands of individuals. All of them include tens of thousands who got the vaccine and tens of thousands who got a placebo, and then they are followed. They're followed with both blood tests for the development of antibody and the persistence of the response to the vaccine, as well as those who may get infected. 2 of those trials have completed enrollment of 30,000 persons. Now that's the number that's necessary. And according to the guidelines from our Food and Drug Administration, once a trial enrolls 30,000 people, the clock starts. And then those 30,000 have to be followed for 2 months. And then it can be submitted to first the NIH and then the FDA. We now have 2 companies who have achieved that endpoint, Moderna and Pfizer-BioNTech. And since they completed it, we're now in that 2-month follow-up period with planned submission to the FDA by the end of November. Very exciting to see what the FDA feels about the trial. So they will be comparing those 30,000 persons who got the vaccine. Looks like I missed a 0 when I typed that. It's 30,000 persons each, not 3,000. And they will be comparing the 15,000 who got the vaccine with the 15,000 who got the placebo. It's also worth noting, because it has been in the news and many questions that I have received, what is going on with the 2 trials in which they have been halted because of side effects. And one of them is the Oxford AstraZeneca trial, and the other one is Johnson & Johnson. Both of these trials are on hold. The Johnson & Johnson has described an individual with a medical complication, but there's no further information, but they have halted the trial themselves. The Oxford AstraZeneca trial, we know a little bit more about. This was halted about a month or 6 weeks ago because of an individual in England who developed a neurologic problem called transverse myelitis. It has since been restarted in England, but not in the United States. However, it's this same trial that made the news last week because one of the individuals enrolled in the trial died. Now, I want to preface this before I go to the next slide. The circumstances around the individual who died have not been publicly released by either the Brazilian Health Service or the company that has the vaccine. The information that is being released comes from the Brazilian news, yet it has not been contradicted. and it has not been claimed to be false. Now, when I do something like this, and we do this frequently at the CDC, we have to be sure that we understand the source and the reliability of the source. So this may or may not be accurate, but it looks pretty good. What you see on the left is that this is the vaccine trial that was halted. It is the Oxford AstraZeneca trial. The person who died was a 28-year-old physician, a frontline physician who treated many COVID patients. He died on October 15th, so died very recently. And he died of complications of COVID infection. He didn't die, according to the Brazilian news, he didn't die from the vaccine, but rather he died from getting COVID. And as you can see, he is an individual who treated many COVID patients. Now, reportedly, and again, this is from the Brazilian news, he was not a vaccine recipient, but rather he was in the control group. The AstraZeneca trial is on hold in the United States because of the previous complication in England, but it is not on hold in Brazil. So this may be true. In fact, I probably would go as far as to say it's likely true, but we don't have a release yet from the Brazilian Health Department or the company. Now, while we're still on the vaccine, I have shown this slide before, but let me introduce it again. There is a nonprofit international organization called One Day Sooner, and the name of the organization comes from, if we can prove that a vaccine works, one day sooner, we will save thousands of lives. So individuals can volunteer to receive a vaccine and then be exposed intentionally to the virus. That is, you get a vaccine, wait 2 weeks for the antibody response, then you get put in a room where they aerosolize the COVID virus. And so far, 39,000 people have signed up for this in 166 countries. Now, this is very unlikely to ever be proven or accepted in the United States due to our human experimentation laws that are actually part of the Federal Register. But that's not true in other countries. In other countries, it is going to be acceptable to voluntarily expose oneself to the virus. And that's what happened this week. This is a picture of a young woman who has in the United Kingdom, and she has posted this picture of herself. Her name is Danica Marcos, and she has received the vaccine, and this week she will voluntarily be exposed to the coronavirus. So this is now been approved in the United Kingdom, but not in the United States. I have no comments about the ethics about it, but I am very interested in the results. And that's about as far as I will go. This is just informative that it is happening in the United Kingdom and likely will happen in other countries, but not here. Now, following that, one of the questions that I get very frequently is, how do I volunteer? No one's called me. There's nothing in the newspaper. No one has asked me if I want to participate in any of the many studies that are going on with COVID in the United States. So there is a federal network, and that is the website at the bottom, www.preventcovid.org. And it lists all of the trials that are going on in the United States and how to volunteer for them if you so choose. And it's not just vaccines. There's many, many other trials going on. All of them are geared to prevention, at least all of the ones to which you might be interested are geared to prevention. So this is how one gets access to the trials if so interested. Now, the last topic, or at least getting close to the last of the topics I want to discuss today, is the United States compared to the European Union. Now, let me start at the top. I don't want the slide to mean versus. I just do that for brevity, but I want to compare what has happened in the United States to what has happened to the European Union and perhaps introduce some stuff that I have not yet discussed. This shows the number of new cases, not total cases, but new cases in Europe compared to the United States. Europe is in blue. So we go back to March and we see that Europe had a big uptick. And this is mostly due to the problems that got wide press in Italy. The uptick in the United States was a little bit later, went a little bit higher, but this is probably true in many countries. There was an uptick around March as we learned more and more about this virus. But what I want to show you here is look what happened in Europe. They went way, way down in the number of new cases, and the United States dropped only a little bit and then is going back up. And so the question is, what's different about Europe compared to the United States? Now, there were theories about this for many, many months until we got involved with Google. Google has a community mobility report. Now, I'm not an expert on this, but it describes the mobility of people that are completely anonymized. That is, they're not releasing any names if they have it, but by cell phone activity, they can create a mobility report. And this is just part of the examples of what they can do. They can look at a population and they have a baseline and then see what has happened. So let's just take the top one, retail stores and recreation. You can see that for this period of time, there was a dramatic reduction by the populace in this area that was sampled of going to retail stores and recreation. Same thing here with grocery and pharmacy. Same thing here with parks. So what it turned out was that in Europe, there was a dramatic reduction of mobility. People stayed home. And I don't know if you recall, but Italy shut its doors nationally. We don't have the ability to do that in the United States, to my knowledge, but that's what happened in Italy, and many other countries in the European Union did the same thing. Now, that's why they had such a dramatic reduction in new cases, and we did not. So the Google Community Mobility Report has now confirmed that that's the reason that there was a reduction in Europe that we did not see in the United States, really based on public activity. And these three topics that I've shown here, for example, are only 3 of 12 things that can be measured. So Google has gotten into this in a big way. Now, as I said, I'm not an authority on this, but having discussed this with people at the NIH, they are, and they feel this is extremely reliable to tell us how much our United States population, as well as population of other countries, are cooperating with recommendations. Now, the next thing is that this report, since it's electronic, can be nationwide, statewide, county, city, and even a large community, such as Brooklyn as opposed to Queens, Manhattan as opposed to Long Island. And it can show you what's going on in various areas. A lot of this information is available just by going on the internet and looking at it, but it is now thought by the NIH to be an extremely reliable index of cooperation with preventative measures. So now let's go back and talk a little bit about the United States and the European Union. I've put these 2 arrows on here because this represents Europe. So we actually stopped the first slide right around here, where the European Union had a dramatic reduction in new cases, and the United States did not have so much of a reduction, in fact, was increasing. Now look what's happened. The United States shows a decrease, and Europe is taking off like a rocket ship. And again, the Google Community Mobility Report shows that this is relaxation. I should say it is consistent with and commensurate with relaxation of preventative measures in Europe. In other words, they're getting tired. We're all getting tired of masking. We're getting tired of not being with family, not going out, not having parties. And this is what has happened in Europe. And unfortunately, as was mentioned earlier, in our own county, we're starting to see an uptick. These are total United States data. They clearly will vary by community, by county, but people are getting tired. And you can see this. So the Google Community Mobility Report is helping us understand this and hopefully helping us to get new guidelines as to what direction we want to go. And finally, at least I think finally, let me show you and emphasize what we do. We don't have a vaccine yet. We don't have a drug that prevents it. So these are the 5 basic preventative measures. If we do these, we can prevent COVID to others and to ourselves. Universal masking or cloth face coverings that we've discussed so many times. Physical distancing is really, really important. At least 6 feet. Now, all of these are not perfect. So yes, it is possible that a shedder of COVID can spread it to someone more than 6 feet, particularly in a closed room with few air exchanges. But these guidelines really will prevent 90% of the cases. Avoid crowds in congregate settings. We know this is going to be tough. We're coming into the holiday season of Thanksgiving and Christmas. The weather is going to change. We're going to be forced indoors rather than outdoors. It's a time for family. It's a time for congregate settings. And all I can say is this is going to be difficult. All of us in the field of epidemiology and science are concerned. And then the last thing is, let's not forget hand washing or hand sanitizing. So Steve, I've got about 10 minutes before 10. Do you want to stop now and take questions, or do you want me to go on for a few more minutes?
If you'd like to go on and wrap up, and then we can stop the screen sharing and then take questions out of work.
Yeah, okay. You know, I didn't know how much time we were going to have this morning. This is another question that I frequently get, and that is herd immunity, and what is it and how do we get there? It actually comes from herds of cattle who had brucellosis, and it was understood then that if the majority of the cattle had already had brucellosis, then the herd was immune. Now we translate that to the cartoon on the right. All of those who are gold are immune. All of those who are red are infected, and those who are green are susceptible. So the green are susceptible, the gold are immune. When one achieves herd immunity, if an infected person gets into the crowd, like any of those red persons, it's unlikely that they will cause a problem. Herd immunity doesn't mean there will be no infections. What it means is an epidemic or an outbreak is unlikely. So we want to achieve herd immunity in the United States. It's impractical to think we will stop this virus completely, but if most people are immune, then we'll stop an outbreak. And the question then comes, how many people need to be immune to create this concept of herd immunity? And what is often not understood, as you can see on the left, is it really varies by disease. So for diphtheria, the threshold is 85%. For measles, we need over 90%. Go down to the bottom for smallpox, only 80%. So the calculation of what is necessary for herd immunity is an epidemiologist's backroom calculation, but it really depends upon how infectious is a virus. What we see on the right is the estimate for COVID-19 is about 60%. And we are not close anywhere in the world. So that's our target right now. Based on what we know about this, if we can get 60% of the people in the United States or in any location immune, we will likely achieve herd immunity. So in concept, currently this is our best solution to a new pandemic. A new pandemic means there is likely no natural immunity. There is natural immunity to many viruses. That means that our human genome makes us immune to a lot of viruses, most of them that are carried by animals. But in a new pandemic, there is likely no natural immunity. We're not close to herd immunity anywhere in the world, but if we get there, New infections will continue, but they will be slow. We've discussed about how do we slow those with the 5 basic preventative measures. And I should say that there has been a very bright group of scientists who have come out with a thing called the Great Barrington Declaration. Great Barrington is a city in Massachusetts, and it creates an interesting controversy. The controversy is focused protection, where we only use the 5 basic preventative measures for people who are most likely to have a complicated disease, and we let the rest of the public run wild. And I don't mean that in a derogative way. What I do mean in science is that controversies are part of science. They're part of medicines. When we don't have the answer, it is common to have opposing viewpoints and then to debate those and look at what is most likely. In fact, for more than 15 years, I have moderated a national session on clinical controversies where we don't have the answers in an Oxford-style debate. Well, I will say that most of the opinions right now are against focused protection. Most of the opinions right now are favoring the 5 basic preventative measures for the public at large. And lastly, it is unlikely that we will see herd immunity in the United States or anywhere in the world without a vaccine. So Steve, that was the last few slides. Let me stop there and we'll turn it back over to you. And Mr. Mayor, once again, thank you very much for the invitation. Always a pleasure to join a panel like this of great community leaders. Thank you again.
Thank you, Dr. Joseph, and thank you as always for giving us the context we need to make smart decisions from day to day to day. Thank you very much.
And I do have one final question that came in, and this one's for Dr. Joseph. With regard to vaccine status, have you heard about the peptide platform that Pharma of Pleasant Hill has developed? Yes.
Thank you for the question. Vaccines are typically 3 platforms. The newest one that is most popular by the 2 vaccine companies that will be submitted to the FDA uses nucleic acid. It's a molecular platform. Both of them are using messenger RNA. The second platform is a virus vector, where you modify a mild virus to produce proteins that will make us immune. And the one that's used most often is an adenovirus. You get a shot with a live adenovirus, and then you get a minor infection with an adenovirus and it produces proteins. And then the third one is just grinding up grinding up the virus and giving particles of the virus, either whole or split or ground up virus, and using that as a vaccine. It's dead virus and looking at the immune response. The new peptide chain is very interesting because it's synthetic. What I mean, peptides are really, really easy to make in a laboratory. And that becomes important. So what one can do then is create peptides that represent either the vaccine spike, which is thought to be the most important part of attachment, or any of the surface proteins, that they're all made up of peptides, and inject that into a person. It has 2 advantages. One is that it's really easy to make and inexpensive, and massive production can be done in months. I mean, you can make 100 million doses in a month. The second thing that makes it attractive is that it's not living, it's not molecular, so it doesn't get into one's host cells, and it creates an immune response. So I think that represents the 4th platform that is exciting, part of what the whole COVID pandemic has taught us, and that's new ways to produce a vaccine. All of that said, it's not in production yet. To the best of my knowledge, it's not into anywhere near phase 3 trials, but it certainly is exciting and worth watching.
And then I have a question for Dr. Joseph, and you've answered this before 3 weeks ago or 5 weeks ago, but I'd like to revisit it, which revolves around, because of Halloween, if children get treats from packaged treats, you had mentioned that is safe for children to receive treats that are wrapped in packages. Is that correct? And is there a period of time you have to wait? What's the usual protocol?
Well, food is an unlikely vector to transmit this virus. So let's start off with the wrapped food. Take something as simple as a candy bar or wrapped hard candy. The food is fine. If there's a risk, it's the wrapping. All right. So let's take the worst case, and let's just assume that this won't happen locally or in the United States. But someone who is a very heavy shedder of the virus sneezes or coughs on the wrapper of a candy bar, and that candy bar is picked up before the virus dies. And then whoever picks it up now has this virus on their hands. That does not cause infection. Causing infection means putting your hands to your face. So that is an extremely unlikely situation, but let's just say it's within the realm of possibility. Breaking that chain anywhere prevents infection. And the easiest way to break the chain is if a child or an adult picks up wrapped candy, when you unwrap it, just wash your hands. Don't put your hands to your face. Just unwrap it and don't put your hands to your face. Unwrap it safely. And then when you take the candy, just be sure you wash your hands. Anywhere along that line will break that extremely unlikely scenario. And the reason I say it's unlikely is so far in the United States, we have not traced any outbreaks to food or wrapped products. So that gives you an idea of how unlikely it is. But again, the common sense, the number 5 of the basic preventative measures, wash your hands.
The RNA can remain on a package for a while, but it's not viable. Is that?
That is correct. So we actually have done studies, many studies, where we will use a Q-tip or a cotton swab a doorknob, swab a laboratory surface, and get those little pieces of the nucleic acid of the RNA that we use for testing. But those viruses are not infectious. So the presence of a positive PCR test does not define infectivity or contagiousness unless a person has been exposed and is symptomatic.
So when you get home, wash your hands.
Wash your hands. Wear a face
Thank
mask.
you. There you go.